Bonjour tout le monde, merci d'être venu pour notre séminaire à l'IBIS. Aujourd'hui, on a l'honneur d'avoir Dr. Kerry Monroe de l'Université de Californie à Davis. Dr. Monroe a complété son doctorat en écologie en 2019 de l'Université de Colorado sous la supervision de Dr. John McKay, où il a étudié l'adaptation climatique chez différentes espèces végétales. Ensuite, il a fait un postdoc au Max Planck Institut en Allemagne, sous supervision de Dr. Dieter Wieger, où il a étudié l'évolution de ratio de mutation chez les plantes. Depuis 2020, il a commencé son labo à UC Davis dans le département de Plant Sciences avec un but d'étudier l'évolution des mutations et aussi leur impact sur le changement climatique et la génomique de plantes. Thank you, Gary, to be here. Thank you for accepting our invitations. We're waiting to hear your nice presentation. Stage is yours.
All right. Merci. Bonjour. That's the best I'll do. Oh, that's all. I have 5 years of French put into action. I wouldn't want to ruin such a beautiful language by attempting to use it here. But thank you so much for the invitation. It's exciting to chat with you guys about some of the work that we've been doing. A lot of this is new stuff, so I'm excited to share it. A lab at UC Davis, we're interested in plant biology, plant genomics, and we work on a number of different systems. So we've started projects in a bunch of different crops. So pistachio, wheat, cassava, celery, walnut, rice. Our work kind of spans a lot of questions in genomics. So we're generating pangenomes in a number of different species. We're asking questions about how that genetic diversity contributes to climate adaptation. And then what I'm going to talk about today is the work about to understand mutation rate as an interesting process in plant genetics. The reason we care is because we know that mutation is the ultimate source of all the genetic diversity that we see in plants or other species. And so understanding the mechanisms that shape why some mutations happen more than others, how those mechanisms evolved, and what the consequences are for the evolution of species genomes is something that we're quite interested in trying to ask these types of questions. So what is a mutation? What are we talking about? So I think this is a fascinating fact that DNA in each cell, this is data from humans, but similar results in plants have confirmed this is also true in plants, is that every day DNA is damaged somewhere between 10,000 and 100,000 times per cell. And this damage comes from many different sources. So we have all kinds of exogenous factors such as radiation, and endogenous factors such as replication errors or reactive oxygen species from metabolism. These introduce all kinds of different forms of DNA damage, which the cell has to figure out how to solve. So most of this damage is going to be repaired, and these are the jobs of all these different DNA repair pathways. And so there's a whole host of proteins that are constantly scanning the genome looking for damage and trying to fix it. But if that fails, if they fail to be repaired, or in some cases, if the repair is kind of inaccurate, then what results is a mutation. So it's really the failure of DNA repair to fix damage, which is what induces a mutation. So for our work, understanding the way in which DNA repair can recognize certain regions of the genome over others becomes very important if we want to ask questions about why mutation rates might vary across the genome. And these can arise in many different forms: substitutions, deletions, inversions, etc. So today I'm going to talk about kind of three different themes. The first is going to be, we're going to be looking at mechanisms of how the epigenome can serve as a can serve to recruit DNA repair proteins via histone reader domains. We'll see how these have evolved, when they evolved, and which ones are unique to plant genomes. And then we'll also take a look at what the consequences of these mechanisms are for the evolution of plant genomes in terms of mutation biases that emerge. But first, we have to kind of address the elephant in the room here about where this work fits within the broader context of how we have historically thought about and talked about mutations. So you've probably heard something along the lines that mutations are random. This is a very common kind of phrase in biology and genetics. And generally what's being described is that this idea that mutations occur randomly, whether because they're independent of whether they're beneficial or harmful. And this kind of language is pretty common in sort of evolutionary genetics and genetic textbooks and so forth, and kind of captures this paradigm that mutation is this random process, and it's through the action of natural selection that we see biases in the distribution that reflect something functionally interesting. And so this is kind of the historical context that we have to kind of be cognizant of as we proceed through these questions. So it's, of course, many of these ideas originated quite a long time ago, and we've come a long way now in terms of our understanding of the actual mechanistic basis of mutation, how it works, how repair works. And what's become increasingly clear is that the mutation rate across organism genomes can be largely affected by the epigenome, so chromatin. We're talking about things like histone modifications and cytosine methylation. All of these things constitute chromatin, and these prove to be very important predictors of things like mutation rate across the genome. So a lot of this work has really been pioneered in recent years in the realm of cancer biology. Obviously, understanding why mutations happen is important for predicting the emergence and outcome of cancer. So quick review of what we're talking about here with chromatin. We're referring to the organization of DNA inside of the genome by these structures. So we have nucleosomes, which around which DNA is wrapped, which are made up of histones. These histones have tails that can be modified chemically. These modifications are established by histone modification writers. They can be removed by erasers, and they are read by readers. So these are all different domains that can be found on a diversity of proteins that can write, erase, or read various histone modifications. So what do we mean by histone modification? We're talking about the language of the histone code. So the 4 variant of histones that constitute the nucleosome octamer are sort of shown here. There's other variants, but these are just 4 different types of histones. But these all have tails, and these amino acid tails can be chemically modified. So for example, if we trimethylate the 36th lysine, we get H3K 36 trimethylation. If we monomethylate the 4th lysine on the H3 histone tail, we get H3K4 monomethylation. I'm highlighting these 2 now because they're going to prove to be very important in the story to emerge about the role that these 2 play in governing DNA repair in different species. So just to give you a nice visual of this, here's an illustration by a student in our lab, Daniela Quiraz, showing the histone octamers and then a tail that emerges, and we can see the 4th this is the 4th lysine of H3, which can be found in multiple different forms depending on how many methyl groups have been added. So if there's 1 methyl group, it's H3K4 monomethylation. If it's 2 methyl groups, it's dimethylation, and so forth. And so these different versions of this lysine are recognized by different types of readers. And so this kind of biology is happening across all of these different types of histone modifications. But I'm showing this because we're going to see how this proves to be a very important mark for understanding plant DNA repair. But before we get to plants, we actually have to to see what we've learned quite a lot. By we, I mean the community has learned quite a lot of about the DNA repair processes that happen in human genomes. There's been a tremendous amount of work in recent years elucidating the mechanisms by which DNA repair proteins recognize certain histone marks in humans. So here's a simple model that's been clarified by a number of different groups and papers over the last decade or so. We know that this protein, SETD2, is a methyltransferase that specifically enriches gene bodies and exons for this mark H3K36 trimethylation. And it's been shown that this mark is then read by a domain that's part of the mismatch repair protein MSH6. The domain is called a PWWP domain. It specifically binds H3K36 trimethylation. Here's an image. We'll look at a few of these kind of images, these alpha fold structures today. So this is the repair domain of MSH6, and here is the PWWP domain. You can see that here in another illustration of the alpha fold structure, where you can see that you have this PWWP domain separated by a kind of an unstructured tail, sort of tethered there, and then the repair protein here. So this is super well studied. Its dysfunction of this system leads to all kinds of problems and cancers. And not only does this MSH6 protein have this kind of system, but there's also been other repair proteins in humans that have been found to have the same kind of targeting. So a PWWP domain in this protein P75 contributes to homology-directed repair. So in humans, what we see is that there's this system of H3K36 trimethylation enriching in gene bodies, and it's responsible for recruiting repair proteins there. I'm showing this, I mean, you know, we're a plant lab, but seeing this is important because it's going to serve as an important contrast to see how plants are different. And so as a consequence of this mechanism, it's been elaborated on extensively by others that regions that are marked by H3K36 trimethylation, which tend to be in exons of actively expressed genes, have a much decreased mutation rate, which has been kind of determined mechanistically to be driven by that targeting of DNA repair. So like I said, we're a plant lab and we want to ask questions about how plants work. And so we've kind of stumbled into this work by starting to ask questions about what the mutation rate is across plant genomes. The way that we go about this in general is to do these kind of mutation accumulation experiments where we start with a plant whose genome is sequenced. We allow mutations to accumulate over many generations while they accumulate through single seed descent. This is important. We're selecting a random single seed at every generation, which means that the efficacy of selection in these experiments is extremely low. So even deleterious mutations should persist, though lethal mutations are obviously going to be undetected. But after many generations of this process and after many parallel experiments according to this approach, we then sequence the genome of the final generation, and then we can simply ask how many mutations emerged. And more interesting to us is where do they occur across the genome? And so we've done this in a number of different experiments in Arabidopsis and asked what kind of features across the Arabidopsis genome predict mutation rate. And what we find is that this feature, H3K4 monomethylation, appears to have, it's the histone modification with the strongest effect, predicted relationship with lower mutation rates. And it's interesting because this histone mark, as we'll kind of elaborate on later, it's enriched inside of essential genes, and we've said we find it in regions of low mutation rate, and it's enriched in gene bodies. If you're interested in sort of the evolutionary theory of some of this, is we found that we could basically model the relationship between H3K4 monomethylation and repair, and we found that this was able to basically selection can easily act on any mechanism that could target H3K4 monomethylation should be readily acted on by selection. So we've gone about and kind of confirmed this again. We've done this. We looked at 10 independent mutation accumulation experiments done by other groups and asked, do we see a relationship with H3K4 monomethylation? And we see this pattern emerging over and over again. We've since looked at a data set in rice. So this is a cool project that you can find on bioRxiv from Daniela Quiroz and others, where we looked at mutations that accumulated in rice that had been subject to fast neutron mutagenesis. And these were previously published and validated to be super accurate variant calling. And we looked at the distribution of these mutations across the rice genome and found this really significant relationship with lower mutation rates being found in regions marked by H3K4 monomethylation in the rice genome. So this kind of suggested that there's some convergent mechanism that's shared between plants by which they target regions bound by H3K4 monomethylation for preferential DNA repair. So we've now been on this this journey of trying to elucidate the mechanism and characterize how it works and when it evolved. So this is the simple model that we're working with. So this has been done. Other groups have established the mechanism by which how H3K4 monomethylation is established, this histone mark. So there's 3 methyltransferases responsible for this. They monomethylate this lysine specifically in active genes. So they are recruited to genes that are actively expressed, and they monomethylate those gene bodies. But for our purposes, the question that is most interesting is what are the proteins that possibly bind H3K4 monomethylation? Because this could serve as the mechanistic basis for this relationship with low mutation rate that we've observed. So we're super excited to see a paper that came out last year where they characterized a domain in a protein called PDS5C. It turns out that this protein is a cohesion cofactor. It's associated with homology-directed repair, so that's already really exciting. And they showed experimentally and with crystallography that it binds, that it contains this tutor domain that has binding specificity for H3K4 monomethylation. So here's the overall protein structure of PDS5, and it's not entirely clear here, but you have this is the functional domain that interacts with other processes, repair, and so forth. And then there's this really long tail that contains this tutor domain. So the way we kind of think about it is that this tutor domain is acting as kind of an anchor that's keeping this protein that's important for homology-directed repair anchored to regions marked by H3K4 monomethylation. Here we can see the results from isothermal calorimetry, which confirm that the tutor domain specifically binds H3K4 monomethylation. And you can see this is the confirmation from the crystal structure, where you can see the monomethylated lysine, which fits perfectly inside of this aromatic cage, which is made up of 3 aromatic amino acids. That essentially the mechanistic basis of the specificity is that it only has room for 1 methyl group, but it needs to have at least 1 methyl group in order to form the binding, in order to bind. And then the reason that it's specific to the 4th lysine of H3 is because it has to also recognize the neighboring amino acid on the histone tail. So we wanted to ask, so given that there's this relationship, does this protein PDS5C specifically recognize regions of the genome where we see low mutation rates? So we looked at ChIP-seq data from PDS5C, and we do find that it tends to target regions of the genome that are marked by H3K4 monomethylation. So in our previous work, we had found that gene bodies have much lower mutation rates than non-genic regions, and we indeed find that PDS5C is targeting these regions specifically. So when we ask where are we finding it in the genome, it's specifically being found in primarily coding regions and introns. We can look directly at the relationship between mutation rate and PDS5C enrichment, and what we find is that regions of the genome that are depleted for PDS5C tend to have much higher mutation rates. And then we can confirm this mechanistic hypothesis is quite clear here, that the regions of the genome that are marked by H3K4 monomethylation are the ones that are being most highly targeted by PDS5C. So there's this really strong correlation between monomethylated H3K4 and the targeting of PDS5C. So this is a very cool kind of starting point. We can identify a mechanism. There's these PDS5 proteins. They have tutor domains that recognize H3K4 monomethylation, and these are known. There's been a lot of work showing that these proteins are involved in homology-directed repair. But what we also realize is that because we've identified a compelling candidate reader domain, this tutor domain that recognizes H3K4 monomethylation, we could set about to ask, are there other proteins in plant genomes that contain similar domains that could also be contributing to these observations of lower mutation rates? So we did something pretty simple. We took the tutor domain of PDS5C, we blasted it across the Arabidopsis proteome. We found 29 genes that had some similarity. We looked at the gene ontologies of these and found that they were enriched for DNA repair processes, which is cool. Of course, 5 of these ended up being PDS5 homologs. So there's actually 5 types of PDS5 in the genome that all seem to have these tutor domains and all seem to be involved in homology-directed repair. But what really struck us as interesting is the observation that MSH6 has a tutor domain, and this is a really central player in mismatch repair and also contributes to base excision repair. This is really compelling because when we know, and this has been established, as I showed before, that in humans, the MSH6 protein has the binding specificity, but it's through a different domain. So it has a PWWP domain instead of a tutor domain. And we've looked at the sequence. These are totally different sequences. They're completely different structures. So it's not the same structure. So this is really cool because it suggests a kind of convergent mechanism where in humans you have MSH6 that has a PWWP, and in plants the MSH6 protein has a tutor domain. So here you can see the structure of MSH6 in Arabidopsis. This is the repair domains, and here is the tutor domain, and it's again kind of isolated from the main repair structure by this long unstructured tail, this kind of tether. So we like to think of it as tutors acting as an anchor that's kind of keeping MSH6 near regions of the genome marked by H3K4 monomethylation. So this is really cool. We have this tutor domain, but of course we really want to ask, is the binding specificity of this tutor domain conserved between PDS5, which has been validated to have H3K4 monomethylation specificity, but we don't know yet whether this is also binding H3K4 monomethylation specifically. So we've kind of set out to answer that question. So here you can see the amino acid sequences of PDS5 and MSH6, and highlighted are above are these little arrows indicate the amino acids that are responsible for the binding of the monomethylated lysine and the neighboring amino acid, and these are known because they were confirmed experimentally to be necessary for that binding specificity. So here you can see this is actually the structure of PDS5 interacting with H3K4 monomethylation. And so we've taken this model and we've been using in silico approaches. So we've been basically simulating the interaction between H3K4 monomethylation and tutor domains using Rosetta and FlexPepDoc, and asking when we generate these simulations, how often do we find this methyl group being bound inside of this aromatic cage? And when we do this with PDS5, shown here, what we find is that it's binding more often with H3K4 monomethylation, and when we compare that to experimental ICC results, we find that basically the in silico modeling is a near perfect correlation with the experimental results done in vitro. I realize that these labels are missing, but this is PDS5C and this is MSH6. Tutor domain. And so when we do the in silico modeling with the tutor domain of MSH6, we find the same relationship. In fact, we may even have higher specificity and higher binding strength to H3K4 monomethylation in MSH6. And here in these little pictures, you can see these are the images of the structure produced in silico of PDS5C tutor domain and MSH6 tutor domain interacting with H3K4 monomethylation. And so what we conclude from this is that we think that MSH6 tutor domain really does specifically bind H3K4 monomethylation. So this provides really strong mechanistic support for this idea that these repair proteins are targeting this histone modification. We wanted to kind of test this experimentally, doing a proper experiment. And so we took I apologize, there's a mistake on this, but we have wild type plants and we have MSH6 knockout plants. And we basically took these, we sequenced them to super high depth. We sequenced to 250x coverage with paired end Illumina sequencing. We used a rather stringent variant calling approach based on this cancer. This is a caller that's originally designed for calling mutations in cancer, where we took every plant and compared it to every other plant in this kind of normal versus tumor approach to eliminate, basically to only include mutations where the mutation is passing every single comparison. So basically, it's a rather stringent approach, but what it reveals is a really strong relationship between mutation rate being affected by this MSH6 protein. So here I'll try to explain these results. So we have this is mutation rates in relation to gene bodies. When we look at wild type plants or heterozygous plants, we see that the mutation rate in gene bodies is reduced by well over 50%. But in mismatch repair deficient lines, this reduction is significantly lower. So there's much more even mutation rate in the mismatch repair deficient lines. Here we have a comparison of the effect of, this is basically the effect size of mismatch repair deficiency on mutation rate as a function of gene bodies versus intergenic regions versus transposable elements. And we can think about this as essentially revealing which regions of the genome are being recognized and preferentially repaired by MSH6 proteins. And we do find that gene bodies are significantly targeted, essentially. So kind of return to the mechanistic hypothesis, we can split the genome up into percentiles based on H3K4 monomethylation, and we do find that regions of the genome that are marked by H3K4 monomethylation are the ones that are experiencing the largest increase in mutation rate in mismatch repair mutants, which is consistent with these being targeted for preferential repair in the wild type. So we're super excited about this, and we're happy to have you guys take a look at our preprint, which is on bioRxiv. We're posting a new version of it, I think today, in fact. So we're excited about that, and you're welcome to take a look at it there. Okay, so to kind of draw this comparison with what I introduced with the human, what we know in humans and what we now know in plants, it appears quite cool example of convergence where you have MSH6 in humans has this PWWP domain. This has been validated to target H3K4 trimethylation. In plants, we have this MSH6 protein. The ortholog of MSH6 has a tutor domain, and this appears to bind H3K4 monomethylation. So it seems like a very cool example of convergence. And we can kind of summarize that here. This is kind of the grand comparison between what's happening in our bodies and what's happening in plants' bodies. That we have this system of PWWP and multiple repair proteins binding this H3K36 trimethylation via PWWP domains. And in plants, it's driven instead by tutor domains that bind H3K4 monomethylation. But one of the things that makes this so compelling and interesting is that in both cases, so in humans, it's known that this mark H3K36 trimethylation, this is found, it's enriched in gene bodies and it's enriched in exons in actively expressed genes. And in plants, it's H3K4 monomethylation is the mark that's most associated with gene bodies in actively expressed genes. And so basically this suggests that in both cases, there's been this kind of evolutionary solution to increasing the rate of repair in those regions of the genome where genes are being expressed, where they're most important. And so this is kind of a yeah, we think it's a very cool example of convergent evolution. So to kind of compare this more broadly, we've basically been looking outside of this simple comparison between Arabidopsis and humans, to ask, how much diversity is there across the tree of life for these repair targeting mechanisms. So this is humans again, and this is plants, and we're focusing specifically on MSH6 in these images. But what's interesting is that we find that fungi, protists, nematodes, most insects, they have an MSH6 protein that has no reader domain. So that's interesting. And then we also find that this PDS5 tutor domain is unique to plants. So we're already starting to see some examples of, there seems to be this kind of diversification that we previously didn't know about, where different organisms have acquired different reader domains or don't have them at all, for preferentially targeting certain regions of the genome for repair. So if we look at this across the tree of life, this is the MSH6 protein specifically, and we've basically taken, this is data, this is somewhat preliminary. This is only 600 genomes, but we've now expanded this analysis to include over 6,000. But it's like impossible to show that on a slide, basically. But the conclusions are very robust when we expand into this larger set. But what we're finding is that we take the MSH6 protein and we've annotated whether or not they have a tutor domain, a PWWP domain, or no domain at all. And what we find is that the tutor domain appears to be unique to the land plants lineage. So it emerged sometime around 500 million years ago, which is approximately when land plants first evolved. So this includes everything from all the way down to like mosses also have these tutor domains. So this is a super ancient mechanism that has been presumably shaping the evolution of plant genomes for 500 million years. And on the other side of the tree of life, we find that the PWWP domain has a interesting but somewhat more complex evolutionary history. It's shared among all vertebrates, which are all here. And then we see some of the basal lineages. So we find like cnidarians and some arachnids have this PWWP domain, but then the insects and worms seem to be lacking it. So there seems like there's either multiple independent origins or there's been multiple independent losses. And so we're still kind of working out the precise evolutionary history here. But what we think is most interesting is that we have just this clear example of, two different sides of the tree of eukaryotic life that have evolved independent repair targeting systems. We're interested in sort of zooming in on the plant lineage, this tutor domain, and asking, how conserved is this domain? And what we find is that this is a very limited amount of the data that we've looked at, because we have hundreds of plant genomes now to compare. But what we find is that it's the H3K4 monomethylation binding residues that are the most conserved amino acids in the tutor domain of this protein. This is sorry for the latency in the image, but this is an alignment of the tutor domains of all of the MSH6 tutor domains across all plants that we've looked at. And by looking at where there's conservation of shape, we can make inference about how conserved and how constrained these regions are by natural selection. And what we find is that it's the amino acids that are the most relevant for binding H3K4 monomethylation that we find the strongest conservation of sequence and structure. So this suggests that there's been, 500 million years ago, this tutor domain was acquired in MSH6, and ever since it's been under really strong selection to have H3K4 monomethylation binding affinity. So if this is the case, if there's really strong selection for this, seems like a really adaptive trait potentially. This raises the question: why is this so important? Why is H3K4 monomethylation recognition by tutor domains and repair proteins? Why is this happening? And why is it different than animals, for example? So we can try to address this by looking at where H3K4 monomethylation is found in plant genomes. And this is well known that H3K4 monomethylation is kind of considered, this is the gene body mark in plant genomes. So here we can see, I apologize, this is supposed to be H3K4 monomethylation here. This is H3K4 monomethylation distribution. And what we see is that it's highly enriched in gene bodies. In fact, this is the mark that's most enriched in coding regions of any of the histone modifications that we've looked at. We can even quantify this in this ChIP-seq data that I showed earlier about PDS5C, this other protein that has a tutor, and we can find that it's highly enriched in coding sequences. We know that coding regions are vulnerable to mutation. These are places in the genome where mutations are more likely to be deleterious. And so having the ability to preferentially reduce mutation rates in those regions, we propose is likely to be an adaptive trait. We can sort of try to visualize this again. Here, this is the sort of cartoon structure of a plant gene, and we find this is just illustrating that H3K4 monomethylation is the mark that's most highly enriched in gene bodies. So this raises a question: what is, if H3K4 monomethylation is found in gene bodies, if it's preferentially repaired by all these proteins, we should be able to observe that result in mutation rates. And indeed, when we've done analyses, this is a meta-analysis of a bunch of independent mutation accumulation experiments done in Arabidopsis. And the divergence from this dashed line represents the reduction of mutation rates in gene bodies over what we would kind of expect by chance. And in all cases, there's this universal observation that mutation rates are lower in gene bodies, which is consistent with their being targeted for repair by these mechanisms. The interesting and obvious exception to this is there was an experiment done with MSH2 knockout lines, and they did not find a reduction of gene body mutation rates. And this makes a lot of sense to us because MSH6 functions as a dimer with MSH6, which would eliminate this interaction between mismatch repair and H3K4 monomethylation. So that is another cool kind of experimental confirmation that we think can be explained by these potential mechanisms. So it's not just Arabidopsis that we see these kind of patterns. We've looked at a bunch of different species. So this is data from Arabidopsis. This is the visualization of this reduction of gene body mutation rates. But we've also done this in rice. So I talked about rice data earlier, and we find the same reduction. We find that gene body mutation rates are significantly reduced in rice genomes. And then we've also been exploring mutations that have accumulated in walnut tissue culture over the last, there's a 30-year experiment with tissue cultures in walnut, and we've been looking at where mutations have accumulated there, and we find basically a very similar pattern. So what suggests is that these mechanisms of repair are indeed leading to a reduction of mutation rates inside of gene bodies, which, given that coding regions are subjected to more deleterious mutation, could provide some selective advantage. But besides just looking at genes versus non-genes, we've also compared different types of genes. So this is data from rice, where we asked, some genes are marked by H3K4 monomethylation and some are not. And we find that even when we only consider synonymous mutations, this is important because one of the things that comes up in this work is, are you really just observing selection? So here we can eliminate that possibility by focusing only on synonymous mutations. And what we find is that there's a reduction of mutation rate in genes that are marked by H3K4 monomethylation. But more interestingly, from a kind of evolutionary perspective, is that what this means is that genes that are actively expressed have a lower mutation rate, which is compelling because genes that are expressed are doing things and are therefore likely to be under some functional constraint. This is also manifest when we look at natural populations of rice and categorize genes based on the evidence of selection we see in those populations. We find that the genes that are under the strongest evolutionary constraints have a significantly lower mutation rate, even at the synonymous mutation rate level. So what this tells us is that genes that are under stronger conservation and stronger selective pressures are the ones experiencing lower mutation rates, which is pretty cool. So why does this matter? Why is this important? We think it's important because so many of our genetic models are kind of oriented around theories about mutation rate that are thinking about things like randomness. If we think that mutations occur randomly across the genome, if they're just as likely to occur in a gene under evolutionary constraint as they are in one that isn't, this is a very different kind of way of thinking about the origins of genetic diversity than one in which mutation rates can differ between those different types of genes. And the theories around this are intimately linked to mechanisms. So the idea that genes under constraint could experience low mutation rate has been challenged by the argument that there needs to be some mechanism that can simultaneously change the mutation rate of multiple genes. But what we've found is that the ability for DNA repair proteins to recognize histone modifications basically provides a solution to this challenge, because these mechanisms can affect the mutation rates of thousands of genes across the genome simultaneously. So how does this fit with our broader research program? We work on a lot of different crops. And so we're kind of taking this to start asking questions about how have these mechanisms potentially shaped the evolution of crop genomes. So here's some examples of data from, this is polymorphism data. So this is just natural genetic diversity in a bunch of different crop species. But what we find is that there's this pattern of polymorphism that looks remarkably similar to the expected effects of some targeted DNA repair processes. And so we're kind of interested to try to quantify how much of these patterns are driven by the effects of this targeted DNA repair, and how has this shaped crop genome evolution? How does this affect the genetic diversity available for breeding and so forth? So to summarize, we've identified mechanisms by which DNA repair proteins target H3K4 monomethylation, which is found in active genes. We've done experiments to validate that this affects mutation rate. We have started characterizing the evolution of these mechanisms. We find cool examples of independent origins in animals and plants. And then we've started looking at the effects of these mechanisms on mutation biases in plant genomes. There's so many exciting directions that we're hoping to take this. We really want to understand where did this tutor domain come from in MSH6, for example. We have a number of, we're getting data for knockout lines and a bunch of different repair proteins and methyltransferases, et cetera. We're looking at mutation rates of structural variants using long read sequencing, and then sort of theoretical questions about, what are the limits to this type of mutation bias? How can we use it? And then how is this relevant for understanding how plants adapt to changing climates? With that, I will acknowledge my wonderful lab. This isn't everybody, but this is a nice picture of us, including the lab dog. So wonderful group. If you guys are ever out in Davis, we'd love to hang out with you. So thanks so much again for the invitation and your time.
Thank you, Yuri. This is wonderful.